Well, good evening. Thank you so much for coming out this evening for our event on creation and science about creation. We're really happy to have Dr. Wile here with us tonight. And then if you haven't seen the information, tomorrow he will be at the Life Church here in town as well for a seminar in the morning. It will be different from what you have tonight. So if you like what you hear tonight, go. If you don't, go because it might be different tomorrow, and you'll like it at that point, okay? So I am going to introduce Dr. Wile. Tonight there's going to be a presentation followed by Q&A. So if you have some questions, there will be an opportunity to ask those questions either live with the microphone, or if you really don't like that idea, you can text the questions as well, and we'll get those asked. So Dr. Wile holds a PhD in nuclear chemistry from the University of Rochester and a Bachelor of Science in Chemistry. He has won several awards for excellence in teaching and has presented lectures on the topics of nuclear chemistry, Christian apologetics, homeschooling, and creation versus evolution. And real quick, I will tell you, I was homeschooled growing up. This man wrote the curriculum we used for science. He also wrote the curriculum that our school here uses for science, and all the homeschool enrichment programs we have use for science. So he's had a large impact on my life, on many of the students here through writing curriculum as well. So please welcome Dr. Wile.
Thank you. Well, thank you. Thanks for coming out. You never know how many people will come to a talk like this, so thank you very much for being here. So this is what I consider, at least, the most exciting evidence related to creation. In my mind, it's it's an exciting time to be a young Earth creationist because there's so much data coming in that's been confirming the predictions we've been making for several years. And so it's wonderful to see that happening. And so I want to talk about some of that today. And probably one of the most interesting things that happened, it's really in the last 20 years, but nevertheless fairly recent, is there's been a big sea change in the field of genetics. You know, I was taught as fact when I was an unsuspecting university student that less than 2% of the human genome is actually functional. 98% of the DNA that we carry around in our bodies is useless, does nothing. Always sounded odd to me, but it's a fundamental concept in evolution, and it has been for some time. And like I said, I was taught that 98% of the human genome is just useless junk. And this junk is important because if mutations are going to produce new genetic information, you don't want mutations happening on parts of the genome that are actually being used. Because it's being used for a reason, mutations happen to it that could cause problems. So you want a pool of DNA where mutations can happen relatively freely without any consequence to the organism. And then if something wonderful happens, then the organism can start using that DNA. So this junk is supposed to be a source of new genes and evolution. And early on, back when I was just roughly out of grad school, it was thought that most of the genome is so useless we might as well not even study it. So Sidney Brenner, who was at the time the director of the Molecular Genetics Unit of Britain's Medical Research Council, was talking to colleagues about the Human Genome Project, about the idea that we're going to finally map the human genome. And he actually said, you know, we shouldn't map the whole genome because we know most of it's junk. In an article that was written about his views, it says this, quote, "He argues that it's necessary to sequence only 2% of the human genome, the part that contains coded information. The rest of the human genome," Brenner maintains, "is junk, and so there's no reason to study it." And there was a good reason to sort of winnow down what we were studying when we first started thinking about mapping the human genome, because mapping DNA was expensive and hard to do. Well, it turns out an easier way was discovered, and so in the end it became a lot easier to map the human genome, and we decided, okay, we'll map the whole thing, even though we think most of it's junk. So that's good. We ended up mapping the whole thing, and we learned a lot. And of course, one thing we learned is that it's not junk. But even as early as 2000, or as late as 2010, which isn't all that long ago, John C. Advise wrote a book called Inside the Human Genome, A Case for Non-Intelligent Design. And he said the vast majority of human DNA exists not as functional gene regions of any sort, but instead consists of various classes of repetitive DNA sequences, including the decomposing copies of deceased structural genes. So the vast majority of DNA is just junk. It's just useless stuff that's just been sitting around. And in fact, what was at that time the gold standard for evolution, for trying to model evolution on a computer, that program is called AVITA. In order to get evolution to occur at all in the AVITA simulation, you need 85% of the simulated genome to be junk. Because once again, this is where the new genes come from. So you've got to have a big pool of junk DNA in order for mutations to happen freely. Of course, now we know that's just not true. This started back in 2005, a scientific initiative that actually started long before then, called ENCODE, took 1,640 data sets for 147 different human cell types. You might not know this, but your body has a lot of different cells in it, right? So your skin cells are different from your heart, from your nerve cells, which are different from your muscle cells and so forth. And so there are a lot of different cell types in the body, and some of them are only there for a while you're developing and so forth. Well, they were looking at 147 different cell types in the human body, and they looked at, like I said, 1,640 data sets, and they looked at how much of the DNA was actually being read. How much of the DNA were the cells actually reading? And here's the conclusion. Among the many important results, there is one that stands out above them all. More than 80% of the human genome's components have now been assigned at least one biochemical function. And this is back reported in 2012. So in the end, I was taught as fact, 98% is junk. And code tells us that at least 80% is functional. And it's at least 80% because we now know there's even more. Since that time, in 2020, 8 more studies were published by the same group. And they found more functional DNA in human cell types that hadn't been studied yet. So in the end, the human genome is more than 80% functional. Very different from this idea that 98% of it is junk. Dr. Ewan Brenny is the leading data analysis coordinator of this project, and he simply says, we don't really have any large chunks of redundant DNA. This metaphor of junk isn't that useful. But it was something that was very, very much considered fact for a long, long time. I don't know how to pronounce this guy's name, so I'm not going to try. But this guy was on the team that had this results, and he says this, I don't think anyone would have anticipated even close to the amount of sequence that ENCODE has uncovered that looks like it has functional importance. I do. I know a whole group of scientists that thought that. The creationists did. In fact, the creationists predicted it. The creationists predicted that we would eventually find that this fact of biology is absolutely wrong, that the vast majority of human genome is functional. In fact, David DeWitt, who's a professor of biology at Liberty University, he made this video 10 years before the ENCODE project. And this video, if you can't read the title, is Junk DNA is Not Junk. And that was the standard view of creationists. Creationists said that we're going to eventually find that the vast majority of human DNA is in fact functional. Because DNA is designed. If I take something like a car that's clearly designed, I know that 98% of it is not going to be useless. And the same thing with DNA. DNA was designed. It couldn't work if 98% of it was useless. So we know that. Creationists have always known that. Evolutionists have finally caught up. Well, some evolutionists have caught up. And this prediction has basically been confirmed throughout most of evolutionary biology. The Oxford Journal Genome Biology and Evolution stated it quite succinctly. The days of junk DNA are over. Because in the end, the data showed that the creationists were right all along. Now, this is particularly bad for evolutionary theory. For lots of reasons. One, if you don't have a big pool of DNA to do a bunch of free mutations with, it's going to be hard to get any genetic innovation. Dr. Dan Grauer, who's a professor of biology at the University of Houston, has a completely different reason for seeing this is bad for evolutionary biology. And in fact, he sets it quite succinctly. He says, if ENCODE, and remember that's the group that showed that 80% of the human genome is functional, if ENCODE is right, then evolution is wrong. Now he's an evolutionist, but he's at least being honest about this, that evolution requires a lot of junk DNA. Not only as a source of genetic innovation, but as he says, in the end, if you've got if 3.3 billion base pairs of DNA are all useful and subject to mutation, then there's going to be a lot of bad mutations that happen in every generation, mutations that cause problems. And if the human race is going to survive, we can't have all those mutations causing all those problems, so natural selection has to weed those out. And the more functional DNA there is, the more natural selection has to weed out. In other words, the more humans that have to die in order to keep all these bad mutations from piling up. And in 2017, he wrote a scientific paper where he presented his model that basically said, based on the number of human beings that are born every generation, the maximum amount of DNA that could be functional is 15%. In the end, that's the only level of functionality that can be supported by natural selection with the number of people that are born every generation. Now, ENCODE is saying it's a lot more than that. So I absolutely agree with him that if ENCODE was right, then evolution is wrong. Because I think it's a big, big problem for evolution. Now, honestly, I think as time goes on, it's going to be very, very clear that ENCODE was right. And so I doubt that he'll continue to believe this statement, because I expect he'll continue to believe evolution. So he'll find some workaround for it. But nevertheless, right now at least, he's making the obvious statement that's really true. All right? Now, this is important because the creationists made a prediction that's eventually been verified, that the vast majority of human DNA should be functional. Evolutionists made the opposite prediction. The vast majority of DNA should not be functional. In science, in the end, the way you test scientific theories is you have them make predictions. And when the data confirm a prediction, then that lends support to the explanation. So for example, Jerry Coyne, who's one of today's greatest evolutionary evangelists, he says this: for a theory to be considered scientific, it must be testable and make verifiable predictions. And that's actually true. Most people don't understand this. What makes a scientific theory scientific is not that it can explain things. A bedtime story can explain things. What a scientific theory needs to do is predict something we don't know. And then we can go test that prediction, and if the prediction is confirmed by the data, then we can have faith in the scientific theory. At least we have more faith in the scientific theory. And that's what he goes on to say. If those predictions are met, it gives us more confidence that the theory is true. And so in the case of junk DNA, we had two theories. One predicted the vast majority should be functional. One predicted the vast majority should not be functional. We now know the answer. It was the creationist prediction that was correct, the evolutionist prediction that was wrong. And it turns out this has happened so much over the recent couple of decades. Creation theory has made many, many, many testable predictions, and they have generally been confirmed. And so that gives me a lot of confidence that the creation theory is true. And this goes all long before genetics. For example, and this is right in my field, so this is something that I really love. One creationist prediction for years has been there should be a significant amount of carbon-14 in dinosaur bones. Because carbon-14 is a radioactive substance. There are different kinds of carbon, carbon-12, carbon-13, carbon-14. Carbon-12 and carbon-13 are stable. Carbon-14 is not. It radioactively decays. And it actually radioactively decays into nitrogen-14. So if I had a lump of just pure carbon-14 in my hands, over time it would actually look like it was disappearing, because it would turn from solid carbon into gaseous nitrogen. Now, it takes a little time for this to happen. Basically, this happens with a half-life of 5,700 years. And in terms of most radioactive isotopes, that's pretty short. And what a half-life is, it just basically says every 5,700 years, half of the carbon-14 turns into nitrogen. So if I start off with 100 grams of carbon-14, after 5,700 years, roughly the length of this talk, half of it goes away, and I now have 50. If I wait another 5,700 years, half of that will go away, and I'm left with 25. So every half-life, half of the carbon-14 goes away. Well, if you keep cutting the amount of carbon-14 in half, it eventually gets so low that it's not possible to detect it anymore. And the general rule of thumb is after about 10 half-lives, a radioactive substance gets so, the concentration gets so low, we can't detect it anymore. So essentially, carbon-14 has a lifetime of about 60,000 years. After that, there'd be so little of it, it can't be detected. As a side note, if anyone tells you carbon-14 dating indicates the Earth is millions of years old, you can ignore them because they have no idea what they're talking about. In the end, carbon-14 dating can be used, but its theoretical limit is 60,000 years. Its practical limit is about 45 to 50,000 years. So anyway, it doesn't go on for very long. Well, dinosaur bones are supposed to be 65 million years old or older. However, young Earth creationists say no. Dinosaurs were wiped out in the flood. The flood happened only thousands of years ago, so there should be plenty of carbon-14 left in these dinosaur bones. So we have tried to get paleontologists to test these dinosaur bones for carbon-14. And they kept saying, no, why would you? Because it's not going to be there. We know these bones are millions of years old. So in the end, there's not going to be carbon-14 in there. There's no reason to test it. Well, creationists eventually were able to dig up their own fossils and dig up enough contributions to pay a carbon-14 dating lab to actually look at these fossils and see how much carbon-14 was in them. And guess what? The creationist prediction was right. There's plenty of carbon-14 in every dinosaur bone that's been tested. So for example, this is Acrocanthosaurus. It's supposed to be 100 million years old. Fossils from this dinosaur have been carbon dated from somewhere between 23,760 to 30,640 years old. Now, I don't think that's correct. I certainly don't think they're millions of years old. As a nuclear chemist, I can tell you carbon-14 dating has a very limited range, and this is way out of its range. But nevertheless, if those fossils were millions of years old, the radioactive light dating lab should have come back and said, we can't date it, because there's no carbon-14 there. But in the end, there was enough carbon-14 to get these dates. They're wrong, but they at least show there's carbon-14 in the fossils, which is exactly what evolutionists said shouldn't be the case, and exactly what creationists said should be the case. And this isn't just one or two fossils. Here's a Triceratops, supposed to be 65 million years old. It carbon dates somewhere between 24 and 39,000 years old. And in fact, samples of 8 other dinosaur fossils from Texas, Alaska, Colorado, Montana, China, and Europe all had so much carbon-14 date in them that none of them, according to the carbon-14 dating method, should be more than 39,000 years old. Not a single sample tested as expected by evolutionists, which is the result should have been, we can't date it because there's no carbon-14 there. So in the end, here's another example of a creationist prediction being confirmed and an evolutionist prediction being falsified. Now, what would you do if you're an evolutionist? I know what I would do. I need to explain why there's carbon-14 in there, right? If I really believe these are 65 million years old, then I need to figure out a mechanism by which carbon-14 could get into those bones. How am I going to do that? I can do that by testing more bones. And maybe if I test more bones, I can figure out some pattern, right? So what I would think a good scientist would do is want to do more and more and more of these tests. Guess what the evolutionists did? They decided we're not going to do any more tests, because the lab that was being paid to do these tests is an evolutionary lab. And here is a portion of the email they sent to these creation scientists when they found out what the creation scientists were doing with the results. So the lab is called CAIS. And here's a portion of the email that lab sent to these creation scientists. The scientists at CAIS and I are dismayed by the claims you and your team have made with respect to the age of the earth and the validity of biological evolution. Consequently, we are no longer able to provide radiocarbon services in support of your anti-scientific agenda. Let me ask you, what's more scientific? To do tests or to not do tests? Especially when, and I have no problem if you think these bones are 65 million years old. I have no problem with that. But you have a problem in explaining why there's carbon-14 in them. And if you're going to be scientific about it, the only way you're going to solve this problem is to do more tests. And the fact that you're not doing it is almost like you're a little child putting hands over ears and screaming so that you aren't willing to face the facts. And once again, the whole business of science is, if you've got this theory and you really believe it, then that's fine. You should work to support that theory. But you can't just ignore evidence that contradicts your theory. If nothing else, you've got to confront that evidence. That's not what the evolutionists are doing. Spoiler alert, this happens another time, later on. Okay. So speaking of dinosaur fossils, this isn't really a prediction because I don't know of any creationists that predicted this, but this is very interesting. In 2005, Dr. Mary Schweitzer found soft tissue in a Tyrannosaurus rex femur. Now, the femur is the bone that goes from the hip to the knee. In most vertebrates, most animals with backbones, it's the longest bone in the body, not all, but most. Schweitzer had found this bone, but in trying to remove it from the material it was in, they ended up breaking the bone. And inside the bone, they found this. This is a little glob of red, and it's connected by a little bit of tissue there. And in the paper, they say if you take forceps and grab onto this, you take forceps and grab onto this, and you pull, this thing stretches like a rubber band. And when you release, it goes right back to its original size. So this is elastic, soft tissue in a fossil that's supposed to be 65 million years old. Now, laboratory studies say, yeah, soft tissue doesn't last very long. In most laboratory studies, it decays in about a year or so. And even the chemicals that make up soft tissue in most vertebrates, proteins, have a theoretical lifetime of only 30,000 years. So after 30,000 years, the very molecules that make up this soft tissue should be decayed away. And this is supposed to be 65 million year old soft tissue. Now, of course, that's a problem. So initially, evolutionists scrambled, and Schweitzer's an evolutionist, so she thinks there must be some hitherto unknown chemical process that's keeping this tissue soft. But most evolutionists wanted to discount Schweitzer's results because they couldn't see a way for soft tissue to be around for 65 million years. So they scrambled for another solution, and one of those solutions is a bacterial biofilm. So we know bacteria can invade fossils, and when those bacteria eventually die out, they often leave a film behind, and that film can look a little bit like the soft tissue she found. So initially, evolutionists were saying, oh, this is just biofilm left by invading bacteria. But of course, Schweitzer, being a good scientist, said, well, let's test this idea. So I'm going to analyze some of the chemicals that are in this soft tissue. And the chemicals, the proteins specifically, are typical for what we think dinosaurs would make, but they aren't the kind of proteins that bacteria make. So based on the chemical analysis, this is almost certainly not bacterial. Now, since this initial kind of shocking finding, there have been all sorts of examples of this. So this is now well accepted, even among evolutionists, that there has to be soft tissue in dinosaur bones. But during this initial time where there was still kind of some excitement about it, Jack Horner, and I don't know if you know Jack Horner, there are some people who say Indiana Jones was kind of based on him a little bit, although Jones was an archaeologist, Jack Horner is a paleontologist. But he's kind of this freewheeling paleontologist who's discovered a lot of unique fossils and so forth. Well, he runs the museum in Montana that Schweitzer was working in, doing her PhD work in. So in the end, his museum owns the soft tissue and so forth. A creationist group offered his museum a $10,000 grant if he would simply carbon date that soft tissue. Because once again, if it's millions of years, there shouldn't be any carbon-14 in it. If it's thousands of years, there should be carbon-14 in it. Well, Jack Horner actually said that the spin the creationists were going to get off this test is not going to help us, the museums. So he said, no, we're not going to do that. That's why they said, we refuse to do tests if we don't like the results, right? Now, this $10,000 grant was above and beyond the cost of the tests. So the creationists were going to pay for the test and give the museum $10,000 just for letting that test happen. They actually then upped the ante and said, we'll include 4 other artifacts of the museum's choosing. So we'll pay for 4 more artifacts to be tested. And Horner still said no. Once again, if you're a scientist, you would want to do those tests if you're interested in science. But if you're not interested in science, then you're not going to want to do those tests because you don't like what they're probably going to show you, right? Since then, lots and lots and lots of other examples have been found. And one of the most incredible ones, this is a 65 million years old, supposedly, Triceratops horn. It's not this Triceratops horn, because this is a full skull, or mostly full skull. They found just the horn, right? And the horn by itself isn't useful a lot. So they soaked the horn in weak acid to remove the minerals. I don't know if you've ever done this. It's a great experiment to do if you've never done it. Take an egg and put it in a bowl of vinegar and leave it overnight. In the morning, the shell will be gone. But the egg will still be there because there's an organic membrane under the shell that holds the stuff inside the egg. So you have this really, really fragile but intact egg without a shell on it. It's really, really cool because the weak acid gets rid of the minerals but not any of the tissue. That's what they did here. They said, okay, this horn's got a lot of minerals in it. Maybe it's all minerals. What we're going to do is soak it in weak acid, see what's left. What was left was soft brown tissue. That tissue was then analyzed, and it turns out that the analysis showed bone cells. There's basically only one kind of cell in organisms that looks like this, and it's bone cells. Bone cells are these oblong cells that have all these little extensions. They're called filopodial extensions. And they're actually the way the bone cells communicate with each other. Because bone cells actually have to be able to talk to each other, because like when you're like me and you start gaining weight, your bone cells suddenly realize the bones need to be made stronger. So they need to make more bone matrix. And so when the bone cells sense the stress that the extra weight's putting on, they tell each other, let's make more matrix, make these bones stronger, because this guy is getting heavy, right? And so this is how they talk to each other. These filopodial extensions are very, very small. So this black bar here is supposed to represent the length that 20 millionths of a meter would be at this magnification. So these cells are a little over 20 millionths of a meter across. However, these filopodial extensions are very, very tiny. They're much less than even 1 millionth of a meter across. And yet, these thin filopodial extensions are still soft. Here's the way you say it in science speak. Filopodial extensions were delicate and showed no evidence of any permineralization or crystallization artifact, and therefore were interpreted to be soft. So in the end, these cells are soft. Now, they're not alive, they're dead, but they're still soft. So that not only just the bulk tissue is soft, but at least some of the individual cells that make up that tissue are still soft after 65 million years. And even the smallest bit of them, not hard yet, not mineralized. The horn itself was carbon dated, and once again, it had plenty of carbon-14 in it, which it shouldn't have if it was 65 million years old. Now, once again, how do evolutionists respond? Well, the study's principal investigator, Mark Armitage, was promptly fired from his position after publishing this article. Now, I've talked to Mark. He and I have a good collegial relationship, and he says it wasn't the paper that got him fired. He's fairly confident of that. The problem was this. Once the paper was published, students found out that they could go to his lab and look at dinosaur cells. So he said there was hardly a day that went by that he didn't have at least one student coming to his lab asking to see the dinosaur cells. And of course, he would show them why he thought they were soft and everything. And the biology department did not want the students learning that. They didn't want the students excited to see dinosaur cells because those dinosaur cells are not able to be explained right now under the current paradigm, so we don't want students to be exposed to it. He said that's why he got fired. Fortunately, the most vociferous man in the department actually walked into his office and shouted, quote, "We are not going to tolerate your religion in this department," end quote. That was a bad move because, of course, Armitage was able to sue the university for that kind of statement. And the university settled for an undisclosed amount of money. All right? But what he has done with this undisclosed amount of money is that he has actually isolated these cells. He's got them out of the rest of the tissue. This is a microscopic photograph of a dinosaur, a triceratops cell floating in water. You know what that is? That's the nucleus of the cell. He has confirmed using stains that only bind to DNA that there's DNA in that nucleus. So in the end, if you can collect enough of these cells and do the right kind of processing, you can actually start sequencing Tyrannosaurus rex DNA. Not Tyrannosaurus, sorry, this is Triceratops. Now, it's not clear how degraded it would be. It's not clear how useful the information would be. But until you try, you just don't know. Right? Now, this doesn't lead to Jurassic Park because it takes a lot more than DNA to make a living cell. You know, all of the cloning that we do, we start with a living cell, we remove its DNA, and then we replace new DNA in it. But the cell has to be very, very similar to the organism whose DNA you're putting in, or it never works. So in the end, I doubt that we'll ever get to Jurassic Park, but we might at least learn something about what Triceratops DNA looks like. And that would be really, really cool. But once again, so far, it's not the evolutionists doing this work. The evolutionists are firing people who do this work. It's the creationists who are doing this work. And it turns out soft tissue in dinosaur bones is pretty common. So a few paleontologists decided they'd just go through the museum storage lockers where they store bones and pulled out a bunch of dinosaur bones. And one of the principal authors actually described them. He said, quote, "They're very scrappy, individual broken bones. I can't even tell you what dinosaur they came from. And yet, they found red blood cells and proteins, both soft, in those cells. So this is, or in these dinosaur fossils. So this is really common. It doesn't give you a lot of faith that these bones are really millions of years old when all this soft tissue and these chemicals whose theoretical lifetime is 30,000 years are still there, right? And if you really, really want to believe this, here's the current record holder. This isn't a dinosaur, this is a worm. According to the rock it was found in, it's supposed to be 550 million years old. And yet, there's soft tissue in there. After 550 million years. Here's how the authors of the paper said, "Minerals have not replicated any part of the soft tissue, and the carbonaceous material of the wall is primary, preserving the original layering of the wall, its textures and fabrics." In other words, hadn't fossilized yet. Still soft. That's amazing after 550 million years. Right? Now, it's interesting enough if this worm is as old as most creationists think, which is around 4,000 or 5,000 years. But nevertheless, that's a lot easier to believe than millions of years. So in the end, like I said, it's an exciting time to be a young earth creationist. Here's another creationist prediction, the appendix. We all got an appendix, well, some of us don't anymore. But the appendix is this little worm-like sac. It's actually called the vermiform appendix, and vermiform means worm-shaped or worm-like because it kind of looks like a worm dangling off our large intestine. According to a book, and this book is so interestingly named, it's ironic that the title of the book is The Science of Evolution and the Myth of Creationism, Knowing What's Real and Why It Matters. Knowing what's real and why it matters. Here's what it says, quote, "We have an appendix, a small remnant of a prior ancestor's species' intestinal sac, which not only is of no use to us, but which can sometimes kill us when it gets clogged up and infected. What kind of god or other intelligent designer would design organisms with such useless, imperfect, wasteful, and sometimes even harmful features?" I agree, that wouldn't be a very good designer. But of course, creationists have always said the appendix has a function. We just haven't figured it out yet. So basically, the reason everybody thought the appendix was a useless organ in the human body is because in the first world, in the United States, you can lose your appendix and you seem to live an okay life, right? And the tissue doesn't make a lot of sense. It's in a digestive region, but it's full of lymphatic tissue, and lymphatic tissue is infection-fighting tissue. So the tissue in it doesn't make sense for the place it's in. So it doesn't make sense. We can snip it off and still live a good life. It must be useless. Creationists always predicted the opposite. This was back in 1988. Creationist J. Warwick Glover said, the appendix would be involved in the control of which essential bacteria come to reside in the cecum and colon in neonatal life. As well, it would be involved in the development of systemic tolerance to certain anergetic agents within the alimentary tract, whether they are derived from bacteria, foodstuffs, or even the body's own proteolytic enzymes. So if you can't follow that exactly, what he's saying is, it's going to be related to the bacteria that live in your colon. And I don't know if you know this, but in fact you have more bacterial cells in your body than you have human cells. And they're in a lot of places, but they're mostly in your intestines. If you're a healthy human, you have about 1,000 different bacterial species living in your intestines. They digest food that you can't digest. The only reason you can have a good diet of vegetables is because of the bacteria in your colon, because you can't digest cellulose, but they can. And cellulose is a big component of anything that's vegetable. So in the end, the only reason you can eat a good diet of vegetables is because of these bacteria. They make vitamin K for you so your blood clots better, all sorts of good stuff. And of course, you're providing them with food and housing. And so they're paying you for that food and housing with all these things they do. And so he says, look, it's going to be related to that in some way. Well, in fact, the creationist prediction was confirmed. We now know what the appendix is. It is a hideout for your intestinal bacteria. When you have when you have an infection of your intestines, it will tend to wipe out all those good bacteria. And it was originally thought that what you had to do was you had to start gathering those bacteria again. The problem is a lot of those bacteria are gathered when you're eating or drinking your mother's milk. And you don't typically do that after a few years. So in the end, a lot of these bacteria you would be exposed to aren't going to be there if you lose them all to an intestinal infection. Well, it turns out that the appendix is a hideout for these bacteria. When they get the signal that there's an infection, they go into the appendix. The reason the appendix has lymphatic tissue is it's a fort, and it keeps the bad bacteria out. That way, when the infection is finally cleared out, then the bacteria, the good guys that have been hiding out in the appendix, can go repopulate the intestine. And that way you don't have to gather all these bacteria again. They just have to reproduce. Much easier job. Now, the crazy thing is, because of evolutionary theory, it was thought that the appendix was useless, and I am not joking about this. In fact, this is how we found out the appendix was useful. Because of this, when someone was having abdominal surgery and the appendix was easy to access, it was common practice to snip it off and take it out, even if it was completely healthy. That was common practice. Happened to my dad during his first abdominal surgery. Just snipped it right off. This is in fact how we found out, because medical missionaries started going to third world countries and doing abdominal surgeries, and they followed the standard protocol. If the appendix was easy to find, easy to see, they snipped it off. Follow-up studies indicated the people whose appendixes were removed were dying at a much higher rate. And they were dying because when they got an intestinal clearing disease, then in the end, these intestinal diseases can be much more deadly in third world countries. So they were not building up their bacteria enough to fight off the next infection, and so they were dying. And this is how we figured it out. We now know that even in first world countries, this is still a health problem, because we have certain types of bacteria. Clostridium difficile, for example, is still an intestinal infection that happens in the first world. And people without an appendix can never stop getting that infection. They just keep getting it over and over and over because they've lost this special mix of bacteria. People with the appendix, if they have the C. difficile infection, they typically only get it once. The antibiotic clears out, and then the good bacteria come out and repopulate the intestine. So in the end, once again, evolutionary theory, the prediction was, we don't use this thing, snip it off. The creationists said, no, it has something to do with bacteria, better leave it in. Once again, the creationist theory was correct. The creationist prediction was confirmed by the data. Here's another one. This is something, I was an atheist when I first started learning about science. The more science I learned, the harder it was to be an atheist. So I eventually turned from an atheist into someone who believed in some sort of creator God. Now, I know this might come as a shock to some of you, but when I was that age, I was kind of a nerd. Not anymore, but back then, right? And so as a nerd, I did a lot of extra reading. Even though I was an atheist, I wasn't an evolutionist. Because even as a young man first learning science, I saw how evolution doesn't work. So I was an atheist who who decided we hadn't figured out how we all got here yet. So anyway, I was an atheist, reading a bunch of science. Not this particular picture, but this phenomenon was the single most important scientific thing that turned me from atheist into believing in some sort of creator. This is rampant throughout creation. It's called mutualism. Mutualism involves two or more organisms of different species working together so they both survive well. The bacteria in your intestine are an example of that. You're in a mutualistic relationship with all those bacteria. You're helping them survive by giving them food and housing. They're helping you survive by giving you vitamin K and digesting cellulose and things like that. This is a more obvious example. This is a tick bird. And, this can't be really comfortable for this impala, but nevertheless, this tick bird is picking all of the parasites out of the impala's ear. This is food for the tick bird, and it keeps the impala healthier. It happens all throughout creation. As a matter of fact, in the journal Nature, probably, I'm going to say 2010, but it could have been earlier than that, George D. Stanley wrote that symbiosis, the general form of mutualism, is the most enduring biological theme in the history of our planet. More important even than the predator-prey relationship is the mutualistic relationship, because it's everywhere in nature. All right? Now, this is a problem for evolutionists, but this is what I think creation used to be like. When God first created, all of the organisms worked together to make it better for everybody. And in 2003, creationist J.W. Francis said, look, because this is an image of how creation was originally done, then mutualism should be very easy to start, even if these species had never interacted before. So he actually said, if you can find two species that have never interacted, but they each produce something the other wants, evolution will tell you one will become a parasite of the other. Creation tells you they'll work together in a mutualistic relationship. This was actually tested in 2014. In 2014, these two organisms were put together. They had never encountered each other in nature. This lived in one hemisphere, this lived in another. This is an algae, an alga is the singular, and this is a fungus. All right? And we know that algae and fungi do have mutualistic relationships. We call them lichens, so this isn't unusual. But what's unusual is these two guys had never seen each other before. They're both microscopic, and that was done by choice because microscopic organisms are much easier to keep, and they're much easier to study. So in the end, microscopic algae, he makes a chemical that the fungus would like to use. This microscopic fungus makes a chemical that this algae would like to use. So they'd never encountered each other before. They were put in an artificial environment, and in less than a single generation, it didn't even take a generation to do this, a mutualistic relationship developed where they shared their chemicals. And in fact, this relationship was so intimate that this algae formed a shape that had never been seen before in this kind of algae in order to make it very easy for the fungus to snuggle up to it. So basically, the algae made a dent in its outer membrane to allow the fungus to sit right in that dent. It had never been seen, this type of algae had never been seen to make that dent before. But as soon as they got together and noticed they each had something, it made that dent. They made this mutualistic relationship. This is exactly what creationists predict. And it's not what evolutionists predict. Evolutionists predict something else. This book, Symbiosis, An Introduction to Biological Associations, says, quote, "Indeed, it is difficult to conceive of two organisms starting out in mutualistic association." That's not the way evolution works. One wants to survive, one will do anything it can to survive. Most mutualistic symbioses probably began as parasitic ones, with one organism attempting to exploit another one. Then the one that's being exploited develops enough defenses so that in the end the only choice is to share rather than to exploit. That's the evolutionary story of how mutualism happens. The creationists had a different prediction, and once again, it was the creationist prediction that was confirmed by the data. So in the end, if I look at science as a whole, especially right now, I see incredibly exciting data that tend to confirm creationist predictions and falsify evolutionist predictions. As a result, I think it's a very, very good time to be a young Earth creationist. All right, that's all I've got, but we do have time for questions. So we have both live questions, and there was a number you could text. Hope you saw that before.
Yeah, Mason, if you could
put that back up now, because I'm done. Yeah. And I love to answer questions. My favorite thing to do.
So here's the rules. They're very simple. It needs to be a question versus a sermon, all right? And I know you could ask questions about the exact information tonight, or Dr. Wile knows all things creation, evolution, and
Not
beyond.
all things. And I'll be really happy to say...
He knows a lot of things.
I'll be very happy to say the three words that every scientist should be very comfortable saying, and that's, I don't know.
But he'll know he doesn't know, okay? And so if you have a question, creation, evolution related, please just try to make sure it's a question. If you don't want to ask into a microphone, there's the number that you can text. We're going to start with some live questions, though. Raise your hand if you have a question. We have a microphone that Mason back here is going to run to you in order to make sure the people on the live stream can hear it. So while he's going there, I'm going to throw you one that was texted already.
Okay.
Just to get started. What's your number one evidence for the age of the Earth?
Probably my number one evidence would have to be planetary magnetic fields. Planetary magnetic fields, from an old Earth standpoint, you can develop a model that tries to show how a planetary magnetic field develops. However, when you tweak all the fuzzy parameters to make Earth work, then none of the other planets work. You know, as a matter of fact, the old Earth model of planetary magnetic fields said that Mercury shouldn't have a magnetic field. We almost didn't put a magnetometer on the robotic spacecraft that went to Mercury the first time because old Earth people said it shouldn't have a magnetic field. Fortunately, they decided, okay, it's fairly light and fairly cheap, so we're going to put one on there. Guess what? Mercury has a magnetic field. Well, in the 80s, Russell Humphreys produced a model that when you, once again, tweak all the parameters for Earth, it was correct for all of the other planets for whose magnetic fields we knew, but there were two planets whose magnetic fields we didn't know at the time, Neptune and Uranus. 9 years later, the Voyager spacecraft flew by both of them, measured them. His prediction, the creationist prediction, was exactly on the money for both planets. The old Earth evolutionary model was wrong for both planets. For Uranus, it was wrong by a factor of 10,000, which is pretty substantial. That planetary magnetic field model says that the Earth can't be more than 10,000 years old.
How do we know how much carbon-14 was actually on those bones initially?
Okay, so so when we're doing carbon-14 dating, that's a really good question. When you're doing any kind of radioactive dating, I'm measuring the amount of radioactive stuff that's in there now. I have to assume something about how much radioactive stuff was initially there so I know how much decayed, and then I can determine the time. Now, for every method it's different. For carbon-14, it's assumed, incorrectly, that the amount of carbon-14 in the atmosphere has been relatively constant over time. We can do some measurements to show that's not true, but nevertheless, sometimes we make assumptions that aren't true because they're the only assumptions we can make. So we make the assumption that prior to the bomb, because the bomb increased carbon-14 in the atmosphere, prior to the bomb, that amount of carbon-14 was in the atmosphere throughout Earth's history. So anything that died, at least in 60,000 years and before, because that's the range of carbon-14, had the same amount of carbon-14 in it that the atmosphere does now. But once you die, you stop exchanging carbon with the atmosphere because you don't breathe anymore. So in the end, then you have no way of getting carbon-14 in, so then it starts decaying away. Now, we know that's not true, and we can make some corrections for that. So for example, we know that most trees form one ring a year. Some trees do it better than others. It's not perfect in any tree, but bristlecone pines and certain other long-lived trees tend to form one ring a year. When the new ring forms, the old ring dies. So, if I cut down a tree and I count back the rings, I know how old each ring is. So now I can measure the carbon-14 that's in that ring now, correct for the amount of carbon-14 that would have decayed over that time period that we know because we've counted the rings, and I can say, okay, that's how much carbon-14 was in the atmosphere of that year. That's called a calibration. Now we have a calibration for how much carbon-14 was in an animal every year for which we have tree rings. Well, the oldest tree rings we have that we can analyze, roughly 3,000 years. Carbon-14 dating is very accurate from 0 to 3,000 years ago because the tree rings are such a good calibration. After that, we don't have any more tree rings, so we have to go to weaker calibrations, like coral layers. But we know coral layers are notoriously not just one a year, but we still try. So anyway, each section of the carbon-14 calibration gets worse the older it gets. So any kind of archaeological number that you get from carbon-14 that is 3,500 years or less, probably pretty accurate. The older the number gets, the worse it is because the worse the calibration is. Does that help?
All right, raise your hand if you have a question.
Got one over there.
Here and then over here.
Yeah. Love questions.
So will these things help us in the future?
Will this thing help us in the future? Well, I honestly do think, I have another talk that I give called How Evolution Has Impeded Science. And some of it's medical. So for example, evolution has caused people to die because of snipping off the appendix. In the same way, when I was your age, I got my tonsils removed because evolutionary theory said tonsils are useless. As a result of that, I am now significantly more likely to get a respiratory infection than somebody who does have tonsils. And in fact, we know that symptomatic COVID-19 was much more prevalent among people who didn't have tonsils. Those studies have been done. So in the end, I do think it makes a difference because if we stop looking at the world as being thrown together by random processes, but as actually being designed, we do better science. So I do think it can help.
All right. I'm going to ask a text, and then he's coming right here. What are your thoughts on flat Earth theory?
There are more flat earthers alive today than probably ever in the history of the earth. Because please understand, if you were taught that Columbus had trouble getting money for his voyage because everybody thought he'd fall off the edge of the earth, that's one of the many lies, and people knew they were a lie, told anyway, one of the many lies you were taught in school. In 300 BC, Archimedes mathematically derived his law of buoyancy. His first postulate was every surface of water is curved because the earth is a sphere. That's 300 BC. And he didn't make that as a crazy statement. He said, this is well recognized. We know that. In 200 BC, a guy by the name of Eratosthenes used a stick and a 500-mile journey to measure the distance around the globe. He was accurate to within 2% of today's satellite measured value. This is why Columbus had trouble getting money for his voyage. Because of Eratosthenes way back in 200 BC, we knew how far he would have to travel. And no one thought he could make a trip that long without resupplying. And in fact, they would have been correct if it hadn't been for the New World, right? So in the end, ancient people knew the earth, even uneducated ancient people knew the earth was round as long as they lived near the sea. Because there is an ancient Greek term that comes from a Greek mariner, BC times. The English translation is hull down, which means it's so far away I can't see the ship's hull anymore, but I can see its mast. Because the hull is below the curve of the earth, but the mast is not yet. So even uneducated sailors in BC times knew the earth was round. It's absurd that there are flat earthers today. All you have to do is find a friend in the southern hemisphere and ask them to take a picture of the stars one night and then compare them to your stars. They're completely different. Guess what? In 300 BC, that's why Aristotle said the earth has to be round, because he looked at the stars in the northern hemisphere, looked at the stars in the southern hemisphere, and saw they were different. That's impossible on a flat earth. Doesn't take much technology to know that the earth is round. Okay. Oh, there we go.
Doctor, thank you for being here. You make the difficult very simple. I appreciate that. Could you speak to chromosomal differences between apes and humans?
Yeah, so a couple things about chromosomal differences between apes and humans. So for example, the chimpanzee, which is supposed to be our closest evolutionary cousin, I can't remember what numbers that is. It's human chromosome 2, seems to be a merge of 2 chimpanzee chromosomes. So we have one less pair than they do, and it's thought that this is because our chromosomes merged. I think the evidence for the fact that these chromosomes are merged is pretty strong, but it's not explained any better by evolution than it is by creation, because the chromosomal fusion could have happened during the genetic bottleneck that we call the Great Flood, when there was only a few, and if those people had the fused chromosomes, then they would. So I don't think that is good evidence of common ancestry. What most people use as good evidence of common ancestry between chimpanzees and humans, and for example, this is why Michael Behe, who is one of the big intelligent design people, think that we have a common ancestor, is because if you look at certain genes that appear to be broken versions of once working genes, we call those pseudogenes, the mistakes in one of the pseudogenes, the gulo pseudogene, which is used in vitamin C synthesis, the mistakes that occur in the apes and the mistakes that occur in us are the same. There are other mammals that are lower on the evolutionary scale that have working gulo genes. And so they don't have to eat vitamin C, they make it themselves. Chimpanzees, apes, humans, we have to eat vitamin C because we can't make it, and it's thought because it's that gene that got broken. Well, the argument is to get exactly the same mistakes in the ape chromosome as in the human chromosome, very, very unlikely. What's more likely is the apes broke and then gave us that broken chromosome as we evolved from the common ancestor. However, and I agree with that. I agree the odds are long, but there are two things I would say. Number one, we're assuming all mutations occur roughly the same frequency in a gene, and we know that's not true. We know there are mutational hotspots, and so it's not as unlikely because of mutational hotspots. Even then, though, it's still pretty unlikely, and I agree, very unlikely. But evolution requires absurdly unlikely things to happen every step of the way. Creationists only need a reasonably unlikely thing to occur once. I'm going to go with the reasonably unlikely thing occurring once, than absurdly unlikely things occurring time and time and time and time again, which is what evolution requires. So, and it's really interesting that evolutionists use that argument. I can see why they do it, but the fact is they're hoist on their own petard, because in the end they're saying, well, this is so unlikely. Yeah, well, you're wanting proteins to evolve. That's thousands of orders of magnitude less likely, or thousands of times less likely anyway. And you're expecting that to happen all the time. So, does that help? Yeah.
All right, raise your hand if you have a question. Here's one that was texted in. Do you know what the actual age of the pyramids would be?
No, I don't. I know the problem with, and I'm speaking, as a nuclear chemist, even speaking on biology, I'm a little bit, but I've done some research in using nuclear medicine and so forth, so I know a little bit of biology. I wrote a biology textbook, so I know a little bit of biology. When I get to Egyptian history, I'm way outside my comfort zone. But I will tell you something that I've read from many historians, that virtually it's impossible to come up with a unified Egyptian timeline. That pretty much each historian has, not each class, each group of historians have kind of their own preferred timeline, and none of them agree with each other. So I think, given that, I think it would be very hard to put a timeline on the pyramids.
Could you give an oldest possibility if your theory's right?
Oldest possibility of my theory is right. Well, most likely I would say it would have to be post-flood, because the flood almost certainly would have eroded all those pyramids down. And the flood should have happened, 4,500 years. But that's roughly what I'd say the oldest, roughly what I'd say the dinosaur fossils are too, so, yeah.
What triggers a mutation? Is it random, or is there some?
So what triggers a mutation? Is it random or something else? Some of them are random. There's no question about that. All right? But we also know some are not random, because we can make them happen time and time again. So there's a great experiment called the long-term evolution experiment. Richard Linsky started this back in 1984 or 88, I can't remember. And they basically have followed 12 populations of bacteria since that time. They've gone through like 75,000 generations of these bacteria. Every possible mutation has happened at least 3 times. They have never found anything except a bacterium. All right? But they did find a mutation that they thought was random that allowed the bacteria to digest a food that they normally couldn't digest in the presence of oxygen. These bacteria could digest this chemical citrate without oxygen present, but they couldn't digest it with oxygen present. The experiment has oxygen present, so none of the bacteria were digesting citrate, but then 31,500 generations in, one of the populations was able to start digesting citrate. And they traced the mutations that made that happen. They thought those mutations were random. However, Scott Minich and his team, who are one of the intelligent design groups, they wanted to see if that really was a random mutation. So they replicated his results. They found that they could get that mutation to happen reliably, like 80% of the time, in 12 generations or less. So clearly that's not a random mutation. And so that made Minich suggest that there's a class of mutations that the genome is pre-programmed to make, specifically to open up new possibilities. And so we call those adaptive mutations. Those are definitely not random. We don't have a good idea of how much there is. We knew it happened there. There have been a few other papers of other examples that are probably adaptive mutations. But in the end, we know at least some are not random.
I was just curious to know if your studies have ever led you to thinking about life on other planets.
Life on other planets, yeah.
And what they may have on those other planets.
Yeah, so, this is an interesting question because, people talk about life on other planets a lot. And I'm not an evolutionist, never have been. Even when I was an atheist, I wasn't an evolutionist. So I'm not sure I'm exactly imagining evolutionary thinking properly. But if I were an evolutionist, I would never expect to find life anywhere else. Because life is so absurdly improbable that it shouldn't have even happened once without a designer, without any kind of... It shouldn't have even happened once. The fact that it happened once indicates that's the only place it's ever going to happen because it's so absurdly improbable. As a creationist, God created here, he could create elsewhere. So it wouldn't surprise me if there were fish swimming in the methane seas of Titan. That wouldn't surprise me at all, because why would God just limit creating to Earth? Now, maybe he did for some reason. I do think you have problems if I think humans are probably the only organisms in the universe that have the image of God, because you get some theological problems with salvation if there's another population out there with the image of God. But I think in terms of, animal life and so forth, I wouldn't be surprised if there's animal life throughout, and animal life we wouldn't even expect, because God's a lot smarter than us. So it wouldn't bother me, and it wouldn't even surprise me that much. As long as, now, if aliens landed tomorrow and had rational characteristics and could talk about philosophy and things like that, that would bother me a lot. That really would. I have to be honest. But as a scientist, I have to be open to that possibility. And once again, as a scientist, either I have to figure out a way to explain it within my worldview, or I have to change my worldview. Those are my only two choices, right? But as far as animals and so forth, that wouldn't present a challenge to me at all. It wouldn't even surprise me.
All right. Raise your hand if you have a question. I just want to
is there a
remind
follow-up
you.
here? Yeah. Oh, yeah, see, that's the thing. You're saying you would have to have something that's carbon. I'm not sure. Certainly based on the life we know, yeah, absolutely. But God's a lot smarter than us. I mean, we can't make a machine that's 100% efficient, right? But every cell in your body has ATPase, which is a molecular machine that is 100% efficient. Because God can do things we can't. so yeah, I doubt that there's any life like us, because the earth seems so unique in terms of how it's constructed. But it wouldn't surprise me if God created exotic life somewhere else.
I do want to say real fast, because some people might wander out throughout the Q&A session.
Oh yeah, and you should feel free to walk out at any time. I'm a university professor. People walk out on me all the time.
Tomorrow, if you weren't here at the beginning though, tomorrow morning, 9 a.m. at the Life Church, we are going to have Dr. Wile talking some more, but we still have more questions, so we'll go with another live question.
Yeah.
So first question is, can everything be carbon dated, or does it have to be a biological substance? And then second
Can
of
anybody
all,
be carbon dated? Okay, thanks. Okay.
With creation, would you say that all the planets were created at the same time as Earth, or could there, to be still in the creation theory, that other planets could be created before or after?
Yeah. Oh, great question. So first of all, yes, it doesn't have to be living to be carbon dated. Diamonds are carbon dated, for example. Diamonds are, and once again, diamonds, which are supposed to be hundreds of millions of years old, date to about 45 or 50,000 years with carbon-14 dating. So basically, anything that had carbon in it when it was formed should be carbon dated as long as it's less than 60,000 years old. However, there are a lot of chemicals that can absorb carbon from the surrounding throughout time, whether they're alive or dead or whatever. Those can't be carbon dated because there's no way to know. You got to know that they had carbon in them, carbon-14 in them, and then stopped replenishing that carbon-14 at some point. That's the key. Well, a diamond is so hard that the inside of a diamond, you're not going to get anything in. So that would survive. That would suggest. So yeah, you could do it as long as it can't replenish the carbon-14. The planet question is very interesting. Because, we all have this really tidy view of time that was the prevalent view until Einstein came about, that time marches on the same everywhere you go. And I was always skeptical of that because it seemed like in some classes time moved very slowly, and like time might be moving slowly for some of you right now. But in fact, we now know time ticks differently depending on where you are. So, for example, we have these satellites that are orbiting the Earth, and they make our global positioning system work. The only reason the global positioning system works is we take into account that time is ticking faster in the satellites than it is here on Earth. If I were living in the satellites, I would be aging faster than if I were living on Earth. And if we didn't take that into account, the global positioning system wouldn't work. So, the first thing you have to ask when you're asking how old something is whose clock are you using? If you're using Earth's clock and you're a young Earth creationist, you're going to say, okay, the universe is 10,000 Earth years older or less, probably, right? But if the clock is out there, especially towards the edge of the universe, that clock could be reading millions of years, all right? So the universe as a whole was created during that creation week. Clement of Alexandria, who lived in 200 or 300 AD, I can't remember which, actually said it's illogical to talk about time until the end of the creation week because time is a part of creation. And so what was happening during the creation week, it may not even be the same kind of time we're talking about now. But anyway, the universe was created all in that creation week, but individual planets might be formed later in different processes, and they may look a lot older because according to their clock, they are. Does that help? Yeah.
All right. We have a couple Good questions coming in. So the first one, while believing in creation, can the shape of a Neanderthal skull versus a skull from a human today be explained?
Well, first of all, a Neanderthal skull actually has a larger volume than the modern human skull. So if you're going with the naive idea that the bigger the brain, the smarter the organism, which is just definitely not true, but evolutionists use it when trying to do human evolution, then you would say Neanderthals were actually smarter and more evolved than modern humans. Neanderthals, we know, well, we don't know anything in science, but as clear as we can measure, science tells us that humans and Neanderthals interbred. In that case, then, they're humans in every sense of the word. And so in the end, their skull sizes are simply part of the human continuum. And there is a human continuum. I don't know if you know this, but the people with the largest skulls live at the equator. Wait, I got that backwards. Sorry, go backwards. The people with the smallest skulls live at the equator. People with the largest skulls live near the poles. The reason? The farther you are from the equator, the dimmer your world is. So the bigger your optic lobes have to be. So the bigger your brain has to be. So the bigger your skull has to be. So there is actually a continuum of modern human skulls. And we have an average, and the average human skull, modern human skull, is less than the average of the Neanderthals. But the Neanderthals we have, they all live near the poles. So they're going to have big skulls anyway, or far from the equator anyway. So they're going to have big skulls anyway. I don't see enough difference between Neanderthals and modern humans to say that there's any significant difference other than what you find normally among modern humans. And like I said, it's awfully hard to argue that they aren't modern humans since we know we interbred with, that we have Neanderthal DNA in us, which indicates there was some interbreeding that occurred.
All right. Do you believe that dinosaurs cohabitat at the same time as humans? I'm assuming you can decipher that.
Yeah. So I don't think they cohabitated, but they lived at the same time. Yes. And I think there's really strong evidence for this. I've never seen any really solid evidence of human fossils and dinosaur fossils in the same place. I have not seen any strong evidence of that. But there are incredibly accurate drawings of dinosaurs made by ancient people who did not have any kind of paleontological knowledge at all. So, for example, in the Natural Bridges National Monument here in the U.S., you find all these petroglyphs. They were made by the Azenazi Native Americans somewhere between 400 AD and 1400 AD. One of those petroglyphs is a sauropod dinosaur eating a tree. Now, if you're going to imagine some great monster, you might get the sauropod dinosaur shape mostly right, but are you really going to have him eating a tree? Also, it looks like the legs disappear in waves, so he was standing in water, which is exactly what paleontologists thought the sauropod dinosaurs did. They stood in water and ate trees. How did the Azenazi Native Americans know that? Remember, up until the 1600s, there wasn't a scientist on the planet who thought fossils were the remains of once living organisms. Fossils had been found from time immemorial. However, because of Plato's philosophy, it was thought that they were simply interestingly shaped rocks. No, Plato had this idea that there are forms, and forms are what's real. And so biology and geology and physics mimics those forms. So it's not surprising that a rock would grow to look like a snail, because the snail is a form. So the rock will take that form from time to time. That was considered science until about 1600, when Robert Hooke looked at fossils under the microscope and saw the same microscopic structures in the fossil that he saw in the living organisms. That was when we first thought. So even if one of those Azenazi Native Americans had seen a dinosaur fossil, I don't think it would have occurred to him or her that it was a once living organism, because that wasn't the thought back then. I don't know what the Native Americans thought, but nevertheless, that wasn't the thought among scientists anyway. So we have really accurate drawings, and it's not just that one place. If it was that one place, it's a knockoff. Somebody was really lucky. But like in the Havasupai Canyon, there is a petroglyph, a rock piece of art, that looks like Edmontosaurus. There is a bas-relief, I actually have a copy of it. There's a bas-relief in a Cambodian temple that was made in the 12th century that looks exactly like a stegosaurus. Not exactly, there's always art, but looks, anybody looking at it, from a kid who read his first dinosaur book to me, you look at it and say, that's a triceratops. That was built in the 12th century. How did that artist know what a and all the other stuff around it, all the other bas-reliefs are of normal animals, wolves and things like that. But then there's this triceratops. There's a Bishop Bell who lived in the 1500s, Bishop of Carlisle. His tomb has this brass border that has engravings of all these organisms, and they're all just bats and birds and everything, 2 sauropod dinosaurs. How did these people know what these guys looked like? Because I think they were passing down oral histories of what people had seen. They probably didn't live in the same places, but they were probably seen.
Raise your hand if you have a question in person. All right, another one that was texted in, pretty straightforward. How do you know the half-life of carbon-14 is 5,700?
Yeah, this is great because, we have radioisotopes whose half-life is millions of years old. Well, we can't measure them for millions of years, so how do we know? Well, we do this a lot for chemistry. In chemistry, there's all sorts of reactions, decomposition reactions mostly, that are called first-order reactions. They depend only on the amount of one thing. And we can run lots of chemical reactions that occur very quickly, and we can see that all of them follow the exact same mathematical formula when they decay. The only thing that's different is the time constant, the half-life. But the shape of the graph is the same, and they always have the same. The only difference is the half-life, the time. So even though I haven't been measuring for 5,700 years, if I measure the amount of carbon-14 in a sample for, say, a year, and then I use that chemical or that mathematical equation to fit the data that I have, I can come up with the time constant. Now, the longer I watch this, the more accurate my time constant will be. So 5,700 years is a pretty rough estimate. When something's a half-life of 1 or 2 years, we have a very good knowledge of its half-life down to probably a couple of seconds. But in the end, even for something whose half-life is very, very long, if I can just watch it for a while and see what the data look like and then fit those data to the well-established equation, the only thing that's different is the time constant. So that should tell you the half-life.
All right, we have another question here. I'll read a text one though, Mason, while you come to the middle. We'll see if you want to comment on this. I recently saw a study that had studied the DNA of Jesus. It said it was only maternal with 1 extra chromosome.
Yeah, I know. I mean, we don't have any idea of, and we don't think, as Christians, I don't even think you would think that Jesus' material body exists. I mean, he was resurrected materially, and then he was assumed into heaven. So you're not going to study his DNA. If maybe they think they took it off the Shroud of Turin or something, they didn't. Because I was actually part of the lab that developed the method that carbon dated the Shroud of Turin. So I know this stuff really well. And the only thing we could date from the Shroud of Turin was the fibers that had fallen to the bottom of the display case. We could not actually touch the Shroud of Turin to get any sample from it. We had to take the stuff that had fallen to the bottom of the display case. And there wasn't any DNA on it.
Okay, but since you brought that up, do you think the Shroud of Turin has a chance of being the actual burial cloth?
I would say no, but I would say not definitively, because once again, all we could carbon date was the stuff that fell to the bottom. Right? That carbon dated to the 1300s, which is exactly when the Shroud of Turin appeared in history. To me, that's pretty strong evidence that it was something that was, made in the 1300s. However, since we couldn't take multiple samples, and one thing about any radioactive dating technique, even carbon-14 in less than 3,000 years, you take the sample from a different part, you will get a different date. This is one of the problems with radioactive dating is it's not consistent even within the object, right? So since we could only date what little bit had fallen off, I don't even know that that's a really good, accurate date. If I were carbon dating something, I would take samples from all sorts of places and average the result. But we couldn't do that. But I do think it's kind of an interesting coincidence that what we could carbon date ended up being, and when I say we, I wasn't actually doing the carbon dating. I was in the lab that developed the technique. It was another group that was doing the carbon dating, but I was in that lab, so I got to watch it all happen, which was pretty cool.
Just another quick follow-up question to actually just that. Is there other forms of radioactive dating that we can use that can be able to be dated to biological life forms or non-living life forms? And then if there's not, is there another form of dating that does not relate to the radioactive dating?
So there are other forms of dating. There's electron spin resonance dating based on the idea that as molecules decay, the magnetic fields that enter the atomic magnetic fields start disoriented relative to each other. So you could make estimates about how long chemicals have been decaying by looking at the spin resonance. Those are, hard to calibrate, and they don't give us very consistent results. So I don't think they're useful now, but it may be we figure out a way to make them useful. So there are other forms of dating besides radioactive dating that we try and use. Most radioactive dating that we use to date fossils isn't the fossils themselves, because carbon-14 is really the only radioactive dating process that works on the fossil itself. Usually what we do, the method that has the fewest problems, and there's still a lot of problems with it, but the fewest problems for old ages is the potassium-argon dating method, which is only good for something that came from a volcano or magma, so hardened lava. What we usually do is we try and find a lava layer here and a lava layer here, and the sedimentary rock below, in between, is what holds the fossils. The lava layers never hold the fossils because they burn up the fossils usually before the rock hardens. However, if I can potassium-argon date the lower level and potassium-argon date the higher level, that gives me a window that the sedimentary rock layer has to be in between. So if this is 200 million years old and this is 150 million years old, then the sedimentary rock goes from 200 million years to 150 million years. And any fossil that's in there, that's the range. That's how we typically do it, because we can't do the fossils themselves. And most sedimentary rock is very hard to date with radioactive dating because it tends to transport lots of chemicals in and out. So in the end, the lava rock doesn't do that. So that's typically what we do. We call it bracketing. We bracket the sedimentary layers in between the lava layers. The problem with potassium-argon dating is every time we know the age of the lava rock, the potassium-argon dating is wrong. So we know the lava rock from Mount St. Helens. It happened in 1985. It's dated as 12 million years old. There's several lava flows that were recorded in history as being 200 years old, 500 years old. They routinely date millions of years old with the potassium-argon dating method. So if you ask a person who believes in this method, why do you believe it if every time this happens, we know the rock, the potassium-argon date gives us wrong? The answer is you can't use potassium-argon dating on young things. Okay? How do you know everything's not young? I know that's young because somebody saw it in history. But that could have happened at the same time and someone just didn't notice it, right? So how can you arbitrarily say, okay, I can't use it here, but I can use it over here? That's not scientific to me.
All right. Has coal been carbon dated?
Yeah.
And if so, what are the results and where did coal come from?
Yeah, so coal's been carbon dated as well, but I don't think it's a real good candidate for carbon dating because even though it's got a lot of carbon in it, these coal seams are permeated with water a lot of times. And so they're bringing they're bringing, bacteria in, and that's new sources of carbon. So I don't think it's a very good candidate for carbon dating, but people have done that, and it typically carbon dates as being 20 or 30,000 years old. I think coal is almost certainly decayed plants, because plants have a different ratio of the stable carbon isotopes than animals do. The isotope ratios in coal is exactly the same as you find in plants. Also, some coal seams have what suspiciously look like traces of root tracks in them. So I think it's pretty strong evidence that coal is decayed plants. It's thought that oil is decayed animals, and undoubtedly there's some oil that's like that, but there seems to be at least some oil that's also made inorganically. Because in the lab I can simulate conditions, what we think's in the mantle, and I can make oil with that as well. And so most likely oil is a mixture of some organic stuff and some inorganic stuff.
All right, so raise your hand. This is your last chance to lock in that you'll ask a question. Raise your hand if you're going to want to ask a question.
One over there. Got one over
So we have one over here, and then we have one more text. So Mary,
one over here too.
Two more, three more in
We're
person.
locking in. Good. Very good.
Start with Mary.
Okay, I have two questions.
Sure.
So the first one's pretty short. Do they have carbon-14 labs that will, any of them in the world that will use, or are they all basically evolutionary? Or is it just that one that exists?
No, so all of them, basically, in order to do carbon dating the way we do it today, you have to have a particle accelerator. And so particle accelerators are so expensive, you can make them for two things: medicine, which you can charge a lot of money for, or use taxpayer dollars, because that's an infinite supply. Well, for all carbon-14 dating, it's the second, it's taxpayer dollars. And so that's government funding, so that's only going to be done in the evolutionary paradigm. So creationists have to find a lab that's willing to take their money to have dating. Now, I would love to get enough money to actually make a tandem Van de Graaff accelerator for creationists, but you're talking about $2.7 million, and then it costs about $11,000 an hour to run. So it's an expensive endeavor.
Okay, my other question was, you'll read that the magnetic field reverses every however many million years ago or whatever, and they say there's evidence. What is the evidence? And how
There is strong evidence
like
that the magnetic field reversed, because once again, think about igneous rock. When lava flows, it's liquid. When it then cools down and solidifies, it's now a rock. Well, a lot of times you have a lava flow and then another lava flow, then another lava flow. You can find a lava flow where all of the magnetic ores are pointed in one direction, and in another lava flow they switch to another direction, and then another lava flow they go back. Now, the magnetic ores align with the magnetic field of the planet, so that indicates that the planet's magnetic field did flip. Now, interestingly enough, there have been several studies on the seafloor that indicates this happens pretty quickly, like in the order, what they would say is on the order of a couple hundred years. But the old Earth view of the magnetic field says it takes tens of thousands of years for the magnetic reversal to occur. The young Earth view is that all of those flips occurred during Noah's flood, because Noah's flood was started with the fountains of the great deep. And that would have changed the pressure in the mantle, which is underneath the crust, and that would have changed the way the mantle was moving, which is what produces the magnetic field. So when a big low-pressure system occurred because of a big water jet, then you'd have a magnetic reversal until that pressure was relieved, and then it'd go back to its normal way again. So according to Humphreys, or according to the creationist model, that all happened during the flood. And that's at least more consistent with rapid magnetic reversals that we seem to see on the seafloor.
You have so much information here, and it's all great. But this is what I think about with our children today in education, things that are going on, what they're being taught. I'd really like to know for my grandchildren, and for even all of us who are older, Is there a book, a video, or something that starts people out that we could gradually, they can learn what you're teaching us here tonight? Is there one that you recommend, especially for our younger people to start out?
Yeah, there are a lot of great books out there. It just depends on how much you want to read. One of the very best books I think that's still fairly recent is called The New Creationism by Paul Garner. He's a geologist, but he talks about more than just geology. He talks about a little bit of everything. And he talks about how, in science, the creationist view seems to be the one that's being supported by the evidence. And so that's a really good general one to start with. If you're interested more on biology itself, I still think the very best book that's been written on biology is Signature in the Cell by Stephen Meyer. And that talks about the design of the cell and how it clearly indicates a designer. If you're interested in the age of the Earth, there's a book by John Morris, another geologist, called The Young Earth by John Morris. So those are some good books to get you started. And they're all written for the layperson. The Signature in the Cell may be the hardest of those three to read, but they are all written for the layperson. Paul Garner, Garner, Paul Garner.
All right, I have one more text question just to confirm. There were no more in-person questions. We think the third one was itching or scratching the head, so we're going to give that one up.
Okay.
This is follow-up from an earlier question asking, were you saying that Neanderthals were a different race or species? What is their timeline relative to Jesus' life?
So they're not a different species for sure. And I'm not a big fan of the term race, because if I take the average person, let's like typically we say black people are one race, white people are a different race. I categorically reject that. But if I take the average black person and the average white person and I look at their characteristics, they are actually more similar than two randomly chosen people who are white or two randomly chosen people who are black. The averages between the races are more similar than the distributions within each race. So I don't think race is a good term to use. I think we're all just humans. And the fact that Neanderthals, we are almost certain interbreeded with modern humans, indicates Neanderthals and modern humans are the same species. And I wouldn't say a different race, I would just say a different category of human being, because we have all different categories of human beings, just depending on culture and all sorts of different things.
All right. Well, if we have no more questions,
thank you all for your patience and for great questions and for not preaching. Well done.
Thank you, Dr. Wile, for being here. Thank you all for coming tonight. Again, tomorrow, the Life Church, 9 a.m. If you want to come hear some more material, hope to see you there. Have a blessed evening.